Hi, my name is Caitlin Davidson, and I am a freshman this year at the ASU Tempe campus, and my major is biomedical sciences. In Bio 182 this semester, some of the topics we've discussed have been Mendelian genetics, environmental niches, animal behaviors, and many others. The title of my presentation Is There a Gay Gene? And the news story I chose was the article, There's No One Gay Gene, but Genetics Are Linked to Same-Sex Behavior, New Study Says, with genetic study called Large-Scale JWAS Reveals Insights into the Genetic Architecture of Same-Sex Sexual Behavior. For this project, I picked a topic that has to do with Mendelian genetics. I picked a news story about determining whether or not there is a gay gene that can determine if someone is homosexual. The article that I chose is an article from the Washington Post by Lindsay Beaver. This article by Lindsay Beaver is demonstrating that from the evidence of a genetic study called Large-Scale JWAS Reveals Insights into the Genetic Architecture of Same-Sex Sexual Behavior. In the article, Beaver talks about how in the study, data was gathered from around 470,000 people in the United States and in the United Kingdom by a mouse swab they sent in and then filled out a questionnaire about their sexual preferences to determine if there were genetic markers linked to same-sex behavior. When this was done, 26,000 of those people filled out that they had sexual experiences with the same sex. When looking at the DNA of those 26,000 people who had those same-sex experiences versus those who did not, they found 5 genetic variants that were statistically associated with same-sex behavior, but none of them had large enough effects to predict same-sex behaviors. At the end of the article, Beaver talks about how same-sex behavior is a lot more complex than a gene predicting whether or not someone will be gay. The environment is a huge factor in determining same-sex behavior that is oftentimes forgotten because a lot of people think that being gay arises in your genes and only in your genes. The primary article for the news story was called Large-Scale JWAS Reveals Insights into the Genetic Architecture of Same-Sex Sexual Behavior by many researchers, with the lead of the study, Andrea Ghana. In the beginning of the study, Ghana states that only 2 to 10% of individuals engage in same-sex behavior at least once in their lifetime, and that there have been studies in the past that hint at being gay partially comes from genetic influence. In the study, they got data from exactly 477,522 people from the United States, United Kingdom, and Sweden. Once the results and questionnaires have been completed, the researchers found 5 autosomal loci that were significantly associated with, not predicted, with same-sex behavior. In the study, the people that filled out that they had engaged in same-sex behavior all tested genetic variants accounted for 8 to 25% of variation in male and female same-sex sexual behavior. Additional analyses suggested that sexual behavior, attraction, identity, and fantasies are influenced by a similar set of genetic variants. However, the genetic effects that differentiate heterosexual from same-sex sexual behavior are not the same as those that differ among non-heterosexuals with lower versus high proportions of same-sex partners, which suggests that there is no single continuum from opposite sex to same-sex preference. In the end of the study, Ghana had concluded that same-sex sexual behavior is influenced by not one or a few genes, but many. The study also talked about how there is still many uncertainties remain to be explored, including how sociocultural influences on sexual preference might interact with genetic influences. The overall takeaway from the study is that there are genetic variants that associate with same-sex behavior, but that is definitely not what determines being homosexual or not, because there are a ton of factors that go into it. The graph on the slide is a diagram from the study that shows that the diamonds in the red represent genome-wide significant signals from analysis of males and females combined. The triangles represent genome-wide significant signals that are female, pointing up blue, or male, pointing down green specific. To determine if the news article I chose is credible or not, there are many things to look for to make sure of this. First of all, the article is published from a very well-known and well-respected source, which demonstrates that this is trusted and credible information. Another way to determine if a source is credible is if the article used citations to cite other sources and studies like the news story executed throughout the article. One last way to check if the source is reliable is to make sure that the information is recent and up to date, and the article I chose was from 2019, which is still considered credible information. To ensure that I was using the correct steps to identify if my article was credible, I based the rules of credibility off a website from Henry Bull Library. To determine if the primary article was also credible or not, I looked for the same three things as the news article, and the primary literature article exhibited the same accuracy and credibility that the news article does. This slide is showing the credit roll for the images I use in this presentation and video, and the next slide is showing the credit rolls for the sources of information used for this presentation and video. The only person who contributed to this presentation was myself, Caitlin Davidson, in Bio 182, General Biology for Biomedical Sciences, Arizona State University, second semester 2022. Thank you.